Hi, everyone. My name's Allison. I'm an instructor in the Division of Infectious Diseases, and I'm part of Sharon McLaurian's group at Icahn School of Medicine at Mount Sinai in New York. And today I'm going to be talking about our project, a human iPSC Derived Microglia Xenografted mouse model to track CNS HIV infection and test therapeutic interventions. The central nervous system is seeded with HIV during the first 1 to 2 weeks of acute infection. It is thought that infection starts in the periphery where CD4 positive T cells are infected with HIV, which are then trafficked to the central nervous system, crosses the blood-brain barrier, and infects the CNS resident cells. And there, the microglia are the primary target of HIV infection. And infected microglia then causes a downstream pro-inflammatory signaling cascade that leads to neuronal injury, neuronal excitotoxicity, thought to be responsible for HIV-associated neurocognitive disorders. Research on CNS HIV infection has historically been limited due to restricted access to human tissue, especially in terms of performing in vivo studies. This limitation has been circumvented somewhat by the introduction of induced pluripotent stem cells, or iPSCs, where several research groups demonstrated the utility of iPSC-derived microglia to study neurological diseases, both in vitro as well as in vivo using humanized mouse model. And these studies have demonstrated that iPSCs provide an abundant renewable source of microglia, and that iPSC-derived microglia are transcriptionally and phenotypically similar to homeostatic primary microglia, giving them biological relevance as well as translational validity to perform experiments. So our group took advantage of this and developed some models that are iPSC-based for generating in vitro cell culture model as well as in vivo humanized mouse model to study CNS HIV infection. We adapted a protocol that was developed by the Blurton-Jones group at UC Irvine, where we first take the induced pluripotent stem cells, or iPSCs, differentiate them into hematopoietic progenitor cells, or HPCs, which can then be further differentiated into microglial cells. And this entire process takes a little over a month. And each differentiated cells can be validated using cell-specific markers and tested on flow cytometry. So for example, HVCs express CD34, 43, and 45, and iPSC-derived microglia express CD45 as well as CD11B and P2YR12. The HVCs can also be used to develop a humanized mouse model, which is what we did. So we took the HVCs and injected them directly into the neonatal mouse brain. And we used a transgenic mouse that expressed at least one allele of human CSF1 gene that allowed the HPCs to differentiate into microglial cells in the brain, forming a xenograft. And the beauty of iPSC is that they're highly versatile, such that they can be genetically modified. In our case, we took advantage of the AVS1 locus, which in literature has proven to be a safe harbor locus that allows a stable, robust expression of transgene. So at this AVS1 locus, we inserted a Cre recombinase dependent dual fluorescent cassette, where at baseline, iPSC-derived cells would express DS-Red, but when these cells are infected with HIV that expresses Cre, some of these cells would then express GFP. So we call this system HIV-induced lineage tracing, or HILT system, because we can track all of the cells that have ever been HIV infected using this system that permanently marks the cells so that we can trace them at a single cell resolution. So we went on to validate our model. So here you're looking at a picture of the cell iPSC-derived microglia that were differentiated in vitro. So uninfected cells express DS-Red, no GFP is seen. But once these cells are infected with Cre-expressing HIV, some of these cells then start to express GFP. We observed a dose-dependent HIV infection as well as DS-Red deletion in vitro, where once the cells were infected with Cre-expressing HIV, the DS-Red is deleted such that it generates a shorter fragment, and we saw dose-dependent deletion. 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 Next, we went on to validate our in vivo humanized mouse model. So again, the HVCs are directly injected into the neonatal mouse brain, and we let the engraftment go on for 8 weeks, and after which we observed diffuse engraftment of DS-red positive cells, predominantly in the cortical region, as well as some cells in the hippocampal region. This is a half hemisphere of a coronal section of a mouse brain. When we stain these brain sections with human-specific nuclear marker Ku80 and microglia-specific markers IBA1, MP2, RY12, we saw that the DS-red positive cells co-localized with all of these signals, indicating that the DS-red positive cells were human microglial cells that had been xenografted into the mice brain. Next, we went on to test HIV susceptibility of the mouse model. So in order to do this, we inserted an additional engraftment step where we injected the mouse with human PBMC at around week 6 to 10. And here the human PBMC served as a medium for HIV infection, such that when we infect the mice peripherally, the human PBMCs would get infected, and infected cells would then travel to the brain and infect the microglia xenograft. And this model mimics the physiological progression as we know it of the CNS HIV infection, as I described earlier, where infection begins in the periphery and spreads to the central nervous system. Once the PBMCs were injected, we then monitored PBMC engraftment at a weekly basis. So at around 4 weeks post PBMC injection, we observed from 10 to 20, at most 20% of PBMC engraftment in the periphery, at which point we would then infect the mice with HIV peripherally. And this was when the mouse reached about 3 months of age. Once the mice were infected, we then let the infection go on for about 4 weeks, and we then collected the brain. And here you can see that, again, we see engraftment of DS-red positive cells in the mouse brain that co-localize with the human nuclear antigen. And when we stained the sections with HIV P24 antigen, we saw a characteristic cytoplasmic halo around the nucleus, as well as generation of multinucleated microglial nodule in the HIV-infected mouse brain tissue, which we also have historically seen in HIV-infected human tissue. And in the uninfected mouse brain, we did not see any HIV P24 signal. We also confirmed HIV infection using a second method. Here we use an HIV GAG-POL-specific oligo probe in order to perform RNA fluorescent in situ hybridization, or FISH, of HIV-infected brain. And again, we saw expression of DS-red positive cells, as well as expression of HIV viral RNA in the infected mouse brain tissue, whereas in the uninfected mouse tissue, we did not see any expression of the viral RNA. We then went on to quantify the level of HIV infection rate in the xenografted microglia, and we observed that the level of infection in the xenografted microglia correlated with the peripheral HIV viremia using qPCR. And we also quantified the level of HIV viral load in the mouse brain tissue as well, and that also correlated with the plasma viremia in the periphery. We were also curious whether HIV infection induces an immunological response that we would anticipate based on clinical as well as experimental data that had been performed before. And we observed, and for this, we measured the expression level of TNF-alpha and IL-6, both of which are pro-inflammatory cytokines, as well as CD68, which are myeloid cell activation marker. And as expected, the mice that had the highest HIV titer had the highest expression level of all those markers. So in conclusion, iPSCs are highly versatile with potential for genetic engineering, allowing in vivo interrogation of human genetics. In our case, we've developed a transgenic dual fluorescent reporter iPSC model, which allows HIV-induced lineage tracing or HILT system. The beauty of our model is that we can track this frequency and the kinetics of HIV infection in the mouse brain, as well as interrogate CNS HIV pathogenesis at a single cell resolution. And we're planning to perform some 10x genomic studies associated with this. We're also interested in testing molecular, epigenetic, and pharmacological interventions in vivo in humanized mice targeting CNS HIV reservoir and the development of novel therapeutic paradigms. And with that, I'd like to thank all of the members that's contributed to this work, my research advisor, Talia Schwartz, Benjamin Chen, Shram Akbarian, as well as individual members of each of the labs, as well as our collaborators, Samuel Marr's group, the DeWitt Lab, the ID Fellowship, as well as my funding at NIDA. So thank you.
Great, thank you. We have time for a couple of questions, and it looks like there's one in the chat already. So it's just asking, can you provide a little more information regarding how differentiation was induced?
Differentiation, sure. Differentiation of the HPCs and the microglial cells. So we used a protocol that was developed by the Blurton-Jones group at UC Irvine. They have a paper that was written by Amanda McQuad. The protocol is actually in their paper, but there's a vendor at some stem cell technology that sells the differentiation media kit. So we use that for our differentiation.
Okay. And then the next question is, are these microglial cells double positive for PTR-Y12 and TME-M119?
The TME-M119, we did test for that, and it is positive for that. And the first one, sorry, what was the first one?
P2RY12.
Oh, yes, P2RY12, yes. They were double positive for both.
Okay. And then another question is, have you seen region-specific HIV positive cells in this model? And what about behavioral studies?
So we did look at the different regions of the brain that were engrafted with the microglia xenograft, but it seemed like the level of HIV infection that we see, it doesn't really differentiate. Like it's not very region specific. And about the behavior studies, we haven't gone that far yet, but that's something that we are thinking about doing going forward.